Guys, right now I'm going to go through the Plasmid paper cutting activity. Usually we would do this in class, but not all of us are going to be in class and not all of us are able to print. So I just figured I'd go through this and make it easy to understand. So what we have is one sheet. We were given lengths of DNA which we cut, or which I cut, and then tied in or taped into a circle. And so this is going to represent the plasmid that is found in a bacteria. So we remember that the plasmid is the non-essential DNA, usually has genes of resistance, et cetera, et cetera. And so this is the plasmid within the bacteria. And the other sheet is another page, had green DNA, and this is going to represent the DNA that is, or the gene that is going to be inserted into this plasmid. So what happens is scientists find the gene that they want to insert into the plasmid, and so we're picturing this. It's in a test tube. We have all of the bacteria. We have all of the DNA. We just have a bunch of DNA in the test tube, and we add restriction enzymes. And we're, in this case, going to add HIND3. And HIND3 is going to cut everywhere it sees AAGCTT. And what it's going to do is it's going to cut right here, so you can see the A, and then I cut the AGCTT off. And this is going to create a sticky end. And so let's do that to the other end. You can see that in the bold we have the AAGCTT, and we're going to cut that as well. So wherever the restriction enzyme sees the AAGCT, it's going to cut here, and it's going to cut in here, and so it has, right here, we're going to create that sticky end right there. And the scientists are then going to have HIND3 working on the plasmid. The plasmid, we're going to use the same restriction enzyme, and we're going to cut in the same way. So in between the A, or in between the A's, and in between the A's again. Here we go. And so we have created sticky ends. So at this point, this plasmid is not in the bacteria. What they're going to do is they've taken the plasmid out, and they have cut it with the restriction enzyme. And then we're going to put that into the test tube with the gene of interest. And they're going to put those in with a DNA ligase. And because we used the same restriction enzyme on both of these, and we left sticky ends, these are going to, on their own, they're going to find one another and base pair. They're going to match. And then DNA ligase is going to sort of glue them together. And so we have then inserted this gene into our plasmid. Then what scientists do is so we're going to be doing this transformation. We're going to have this DNA, we're going to put it back into the bacteria. And in order to make copies of this, we can just grow bacteria, just grow colonies of bacteria that have this DNA in it. And so therefore it has made many copies of this gene. So I hope this helps, and if you have any questions, please ask.